This is laboratory 1718. It's on EEG and reflexes. The reflexes part is fairly complicated, but the EEG we're going to make fairly simple, and we'll kind of go through it. So for the EEG, we'll use the BioPack, and I'll be honest, there's a lot of conflicting information about the EEG and how it looks. So what we would do is we would hook somebody up. You can see the electrode here on our subject, and we have taken a wrap and put it around the head to hold the EEG electrodes in place. In a clinical situation, they would use pin electrodes, right? So they'd actually put them like acupuncture needles into the scalp. Or in the old days, before they did that, they would literally shave your head. I don't think anybody wants their head shaved for laboratory. So we would hook the person up and put a couple electrodes around the ear, a positive and negative electrode, and then there's a ground electrode. And you measure the electrical differences just like the EMG, right? EEG stands for electroencephalogram. So it's brainwave activity. And so you measure the difference between the positive and negative electrode, and you get an EEG. Now, EEG is fairly simple, and it measures the electrical difference between 2 points in your brain. It's actually looking at the cerebral cortex, and there's always activity. So there's always going to be electrical difference between different points in the cerebral cortex. If there's no activity, you'd have a flat EEG, and by definition, you would be dead, right? So what we would do then is we would calibrate, and once we make sure everything was working, we'd record. And we'd have the person with their eyes closed for about 10 seconds, and then open their eyes for 10 seconds, and then close their eyes again for 10 seconds. And then they would close their eyes and do what's called the drastic. The drastic maneuver is when you take your hands and you clasp them in front of you and then pull apart as far as you can. So you want to put a lot of pressure on that. And when you're done recording, we just hit done, and then we would look at a couple things. We would measure the amplitude of the different waves. The 4 waves in the EEG are alpha, beta, delta, theta, and then we would measure the frequency of the waves as well. So one of the things that kind of surprises students typically is, and our data would look sort of like this, is that the EEG tends to get smaller when your eyes are open than when your eyes are closed. And when students think about it for a minute, then oftentimes they can figure out why. Remember that the electrodes are above the ear. All right, and so what we're doing is we're opening and closing the eyes, and so we're not measuring the visual cortex, right? The visual cortex and the occipital lobe in the back. So we're measuring from the side of the brain, the temporal lobe, which does other things like hearing. So what happens is when someone's eyes are closed, their hearing typically becomes more acute because they're trying to listen to see what's going on around them. And so that's why we see it get bigger. If we actually put the electrodes, because it measures electrical difference between 2 points in the brain, the positive and negative on the back of the head in the occipital lobe, then you would see it get bigger when they open their eyes than when they close, because there would be more visual input. Okay. So this is a typical EEG pattern we'd see, and all we did is we took data like this and we've expanded it so that we can see the individual waves. Okay, so this is alpha, this is beta, this is delta, this is theta, and then the idea would be you measure the peak of one wave to the peak of the next, and then you could get a little frequency button here, and that would tell you what the frequency of the alpha wave and beta and delta and theta, and they all have these characteristic frequencies. I hesitate to show you this, but here they are, and we're not going to worry about them. Okay. So that's it for the EEG. We're not really going to talk much else about anything. And part of that is because the EEG does not really um, show you exactly what's happening. It's not really consistent from person to person, and the information is very different. Most of you won't see it again. Maybe if you worked in a neurology lab, intensive care unit, maybe, or a sleep clinic, maybe. And if you worked any of those places, as in the medical field, they would train you in what you had to do anyway. So I don't think there's a lot of time that we should spend on this. And so I'll tell you the things I want you to know for the EEG. You might as well do it right now. First of all, I want you to know that it measures the electrical difference between 2 points in the brain. And if there's no electrical difference, you'd be dead. And I want you to know that when you do the drastic maneuver, when you take your hands and clasp them and pull them apart as hard as you can, then the EEG gets bigger. So we see an increase in brainwave activity when you do that. And really, that's the couple things I really want you to know for this section, for the EEG. Reflexes are a common clinical test we do, and often during a physical or something like that. Reflexes are covered in FOX 3.3. And we're going to look at some of the more common reflexes and some of the different things that they entail. We're going to basically do some of the reflexes in FOX 3.3. We're going to look at the knee jerk, or sometimes known as the patellar tendon or ligament reflex. And we'll also look at what's called the plantar reflex to see if there's a Babinski sign. So there's a couple other reflexes that they look at in terms of FOX. We're not going to worry about them because they're really basically the same functionally as the knee-jerk reflex. They're all called stretch reflexes, and they're looking across a joint. So for the knee-jerk reflex, right, you have the person usually sitting on the edge of a table, leg hanging down straight. Take the reflex hammer and tap between the tibial tuberosity and the patella. And when you do that, that stretches the patellar tendon, and that causes the muscle to stretch, and that sets up a reflex, and the leg kicks out. So we'll look at that in a little more detail. We will do that with and without the drastic maneuver. And basically what we would do in lab is we would do light little taps and try to find a place where we could consistently get a little reflex. And then we hit harder to see if reflexes are truly graded. Remember, we said that one of the things that reflexes have, one of the properties, is that they're graded. It means the size of response depends on the size of the stimulus. So if I give a big stimulus, if I give a hammer strike that's pretty firm, the leg should kick more than if I give a light tap. We're also going to do that with and without the drastic. And we'll see that when you have the drastic, the knee jerk gets much bigger. And we'll talk about that a little bit. We'll also do the plantar reflex. And the plantar reflex is done at the plantar side of your foot. And basically, reflex hammers actually are built to have a little point at the end you hold for that purposes, or you could use probes or things like that. And what you do is you take your probe or end of the hammer, reflex hammer, and run it up the bottom of the foot. And you're looking at what the toes do. And actually, the toes can do a number of different things. They can flex down. They can flex backwards. They can do nothing. And all of those are normal responses. What is an abnormal response is when the toes fan out. Okay, so they fan out, and that's called a Babinski sign. And a Babinski sign is a sign that there's an abnormal reflex. So we wouldn't expect to see that in the laboratory. The only time I've ever seen it in the lab is I had a student that had a severe back injury, and literally, like the day after the final exam, she was going in for major back surgery. They're going to redo her back and put metal rods in it and all sorts of things. You could tell when she walked around, she was in a lot of pain oftentimes. And so on the one side, because she had disc issues, she had impingement of her nerve on the right side of her body. So you could see a Babinski sign when you did it. But most of the time, when in class, we're not going to see it because people aren't functionally going to be walking around usually with that type of injury. We'll talk about what really the Babinski sign means and how general it really is in terms of things. So again, looking at the EEG, it's an electrical brain recording. Again, it's the cerebral cortex interneurons that have electrical differences, and we can record those with our electrodes. We don't have to know the characteristic frequencies or patterns of these waves. We're not going to worry about them. We're not going to worry about the amplitude. We're not going to worry about when's the alpha or beta or delta is bigger or smaller. Just going to skip that. You got enough stuff to worry about. But what I do want to focus in on is the data right here with the drastic. Okay, so this is the size, and this is, actually this data is bigger in amplitude than we typically see, but it is real data from lab. In the alpha wave, when the person's eyes are open or closed, we see, actually when their eyes are open, we see, for this person at least, a bigger response. But when we look at the drastic, where they're clasping their hands and pulling apart, we see it's significantly bigger, right? I mean, this is 6 times bigger with their eyes closed. This one's 6 times bigger. This one's twice as big, or almost 3 times as big, and this one's 3 times as big, the theta wave. So in every wave, we see a bigger response when the drastic is happening. And basically, it just tells us when you're doing that, you have an increased brainwave activity. And that's going to be an important component of when we talk about the reflex with what's happening. Okay, so increased brainwave activity happens with the drastic, and we can see that right here at this bottom row of numbers. They're all much bigger than the rows above them, in their columns of the rows above them. So basically, that's it for the EEG. Again, measures the electrical difference between 2 parts of the brain. If there's no electrical difference, the line will be flat. If it's a flat line, you're dead. And that drastic maneuver, by clasping your hands and pulling them apart, results in a larger reflex, which means there's more brainwave activity. Okay, so you have a bigger EEG as well. So before we move on to the reflex, let's quickly review the reflex characteristics. We did this in lecture in the communication and homeostasis section. Reflexes are involuntary responses, right? They are caused by an adequate stimulus. You need a specific receptor. The response is patterned, meaning it follows the same pathways every time, and it's graded. We're going to test that a little bit in the fact that the size of the stimulus determines the size of the response. There are five components to a reflex arc. You have a receptor, you have your afferent or sensory fiber, you have your integrator, which is almost always a CNS for us. You have the motor or efferent fiber, that's number 4, and then you have the effector, that's number 5. What we're going to do is we're going to apply this general reflex arc to the knee-jerk reflex. So the knee-jerk reflex, what's happening is, again, we take the hammer and we strike the patellar tendon. Okay, between the patella, it's hard to see, but it's right there, and where we insert at the tibial tuberosity here for the patellar tendon, we strike the tendon right there. That stretches the tendon, and that pulls on the muscle, and that stretches the muscle. And this is a stretch receptor, this blue receptor right here around the muscle fiber. And it's sort of like a spring. So when the muscle stretches, that spring changes size. And what it does is it opens channels and ions move into the cell, and that causes an action potential. So this is my receptor. This is number one. When I have that action potential, it travels down the afferent fiber. So here's my afferent fiber. So it goes up through the dorsal root, the cell bodies in the dorsal root ganglion, goes into the spinal cord and synapses on cell bodies of motor neurons in the ventral gray horn. Okay, now there are two things that happen here without getting into too much detail. One is this directly stimulates the motor neuron. That sets an action potential in that. That goes to the muscle, and that causes the muscle to contract, which prevents the muscle from being overstretched and damaged. Okay? At the same time, the signal also gets diverted into what's called an interneuron. Remember, interneurons are neurons that begin and end entirely within the spinal cord. So typically, the interneurons will be green, right? So the red motor, the blue sensory, and the green the interneuron. And what happens in this interneuron is it inhibits this motor fiber. And basically, it allows the hamstring to relax. Okay? And it makes sense that if I want to move my muscle, I have to contract, or at least in terms of extension, I have to contract the quadriceps muscle, but relax the hamstring muscle. So that's what this interneuron does. It allows us to have what's called reciprocal inhibition and have the muscle kick. Okay, so one, my receptor, called stretch receptors. Two is my sensory neuron that goes up to the spinal cord, right? Three is my integrator. That's right here, this little box where the synapse is. That's within the spinal cord. Four is the motor neuron. And five is the muscle that contracts, the quadriceps. That's my effector. Okay. Now, the knee-jerk reflex has a lot of different names. One of them is monosynaptic. It's called monosynaptic because it's only got one synapse. Okay? So in this reflex, it's got one synapse. So that's a monosynaptic. It's also called a stretch reflex. And it's called a stretch reflex because it's caused by the stimulation of stretch receptors. So stretch reflex. Sometimes it's called a spinal reflex. Why? Because it's mediated at the level of the spinal cord. And sometimes it's called a simple reflex. Simple, because it's pretty simple. It's the simplest reflex you can get, where you have the receptor, the afferent, the integrator, the efferent, and the effector. So those are names you typically see associated with this reflex. Now, the receptors are stretch receptors, and not all stretch receptors look like this, but they go around what's called the muscle spindle, and when the muscle contracts, they change their shape. And changing their shape sets up action potentials, which then go to the afferent fiber. To get these stretch receptors to set up action potentials, we have to reach threshold. So we have to give it a big enough stimulus to get this membrane voltage to reach threshold. If I reach threshold, then I trigger an action potential. All right. So when we give a stimulus, right, we have to have the trigger zone. That's what we call functionally the axon hillock, where the axon attaches to the cell body. This is where the action potential arises from. Okay, generally speaking. Now it's a little different in terms of how things work in the pseudounipolar neurons that our cutaneous senses, and in this case our proprioceptors, follow. But the key is, does the cell body reach threshold? And if it does, it'll stimulate an action potential. And if it doesn't, nothing will happen. Okay? So that graded potential, if it reaches threshold, right, causes an action potential you see right there. Now the action potential is somewhat like doing a wave where the first part of the axon depolarizes, becomes more positive as sodium rushes in. That's a cation, so as it comes into the cell, it makes it somewhat positive. And then that excites the next bit, which excites the next bit, and so on. So it moves down the axon. At the same time, we have the refractory period, which is causing the neuron, basically the axon, to reset so that the action potential can't go backwards. All right. Now, in terms of both the afferent and motor fibers, they are myelinated fibers. They're fairly large. And so they're going to have the saltatory conduction, right, where the nodes only have the channels for sodium. So sodium rushes in, and that changes the membrane voltage. But what it does is it makes the action potential go faster. So I generated down here the axon hillock, right? That's called the trigger zone, and it goes very quickly from one end to the other. And so the action potential travels from the quadriceps, right, up to the spinal cord. Now, in the spinal cord, right, when we get to the synapse, that's our monosynaptic reflex, we're going to release neurotransmitter. How we do that is we open calcium channels. Calcium comes in. That's called calcium current. That's actually energy. And that gives enough energy to cause the exocytosis of neurotransmitter. Neurotransmitter travels across the synaptic cleft, binds to a channel on the other side, and that opens channels in the cell body of the motor fiber, and that initiates the action potential if they reach threshold, and that causes the muscle to contract. All right. And so the motor neuron, in order to do that, has to reach threshold as well. Okay. And if it does, then we'll get a contraction. And that action potential is not any different whether it's carried on the afferent or sensory neuron or the efferent or motor neuron. It looks exactly the same. It's an individual action potential, right? Not to be confused with the compound action potential we studied in the frog. And again, it does the same thing, but this time it's going down in the opposite direction from the cell body to the synaptic ending. When we get to the muscle cell, we're going to have the exact same thing happening, where we're going to open a voltage-gated calcium channel. Calcium is going to diffuse in. That's going to provide energy for exocytosis of the neurotransmitter. That's going to be acetylcholine. It's going to bind to those nicotinic receptors we're going to talk about in the end of the ANS section. And that's going to set action potentials on the muscle cell itself, which is going to cause the contraction of the quadriceps muscle. So that's kind of how it works. Just as a comparison, we're going to talk more about the knee jerk in a second, but just as a comparison to some of the terminology I used earlier, the knee jerk, right, is called a monosynaptic reflex because it involves one synapse. But if you look at the inhibition of the hamstring, right, it's called a polysynaptic, which really is just more than one. You could call it a bisynaptic if you wanted, right, because there's two synapses there. So the knee-jerk reflex is somewhat unique in terms of having only one synapse. Most of the other reflexes tend to be a little more complicated. So when we look at what happens, we can talk about graded potentials. And it's the graded potentials that cause or don't cause the action potential. Now, there are two types of graded potentials. And again, this should correspond nicely with the nervous system lecture. The first type is called an EPSP. Okay, so the PSP stands for in this case, postsynaptic potential. So postsynaptic potential. And the E is excitatory. You can also have an IPSP. Down here you can see it. The I is inhibitory. So excitatory postsynaptic potentials, come over here, right? So they would drive us towards threshold. Inhibitory would take us away from threshold. So excitatory, make it more likely we have an action potential. Inhibitory, make it less likely we have an action potential because we're going away. So we need to understand what an EPSP and an IPSP is for a couple reasons. What level of the spinal cord this occurs at is about L4, okay? And that'll become important a little later as well. So we cause these EPSPs, these excitatory postsynaptic potential, in this synapse, which then causes the contraction of the quadriceps once we reach threshold. Okay? But in terms of the interneuron, it's a little different. Okay? We don't want the hamstring to contract, otherwise, if both are contracted, wouldn't it be able to have any motion, right? So we have to have an IPSP, an inhibitory signal in the hamstring, to relax it. Okay, so how do we get inhibitory signals? Well, it's pretty simple. An excitatory stimulus would be any stimulus that causes an increase in the membrane voltage, that brings us closer to threshold. And there's 4 ways to do that in the human body. Okay, so an excitatory stimulus would be open a sodium channel. Sodium diffuses in, it's a positive ion. It makes the membrane voltage go up, closer to threshold. We can also open a calcium channel. Calcium's higher on the outside of the cell. We open a channel, it diffuses in, and it makes membrane voltage go up in excitatory direction. If enough calcium comes in, we generate an action potential when we reach threshold. And those two are pretty simple and straightforward. We can also do a couple other things. We can close a potassium channel. Why does that work? Well, potassium's higher concentration inside a cell, it diffuses out. When potassium diffuses out of the cell, it makes the inside of the cell more negative. Right? So what you do to make the cell more positive is you close a potassium channel. It's like, hey, if you want to save money, quit spending as much. Right? So the same thing happens with the potassium channel. When potassium's leaving and I want the cell membrane voltage to go up, I close that potassium channel. I keep those positive ions inside the cell and membrane voltage goes up. The other way to do it is close a chloride channel. Now, chloride's a little different. Chloride is an anion, right? So Cl minus. It's a high concentration outside. And again, a good rule of thumb is to think that everything that we're going to talk about pretty much in this class is higher concentration outside a cell than inside the cell, except for potassium. So if you don't know, take a guess. It's going to be outside. So chloride has a higher concentration outside the cell. And if a chloride channel is open, chloride would come into the cell and make it more negative. So if I close a chloride channel, right, close the chloride channel, membrane voltage goes up, and I have an excitatory stimulus. Well, how do we make inhibitory stimuli? Well, we just do the opposite. So for an IPSP, we open a potassium channel or open a chloride channel, or we close a sodium channel or close a calcium channel. Those create the inhibitory signals. So when we do the drastic maneuver and we clasp our hands and we pull them apart as hard as we can, our brain gets busy, all right? And so what does that have to do with anything? Well, if we measure the size of the reflex is much greater when you're doing the drastic. It's really big. It's really obvious. As a matter of fact, for most of you that go into clinical medicine, when you learn about the reflexes, they'll teach you how to do the drastic. If you can't find a reflex on somebody, you have them do that, and that tends to make it much easier to find. Some people have reflexes that are very difficult to locate. And if they do have reflexes that are not working properly, then that is indication there's something wrong in the body, oftentimes in some sort of ion balance or something. So the drastic makes the reflex larger. And the reason why is kind of complicated, but it's good physiology. Normally, when you do a normal reflex, your brain is signaling down to interneurons. And the interneurons your brain signals are called Renshaw cells. So you see it right here. What Renshaw cells do is they make iPSPs in the motor neuron. Okay? So Renshaw cells are these interneurons that are located, not this one, right? So it looks sort of like this. This is what this one's showing. This Renshaw cell is going to the brain, all right? And this Renshaw cell is making inhibitory signals to the motor neuron. And so it's sort of canceling the neuron signals coming from the sensory neuron, because those are excitatory. Now, it depends on the relative size. If I have a lot of excitatory stimulation and small inhibitory stimulation, I can still reach threshold. But it's helped to basically make this less information by canceling it out, this inhibitory. And the idea of these interneurons and the Renshaw cells being inhibitory is that they tend to smooth out reflexes. They make them less jerky. And so normally your brain is signaling the interneurons, those Renshaw cells, to cause iPSPs. We know now that they open chloride channels in the cell membrane. And as chloride comes in, that keeps membrane voltage lower. And it tends to inhibit the reflex a little bit. The reflex response isn't as big. But when you clasp your hands and pull them apart, we saw in the EEG section that we had a larger EEG when the person was doing the drastic. And that says that we have more brainwave activity. And basically what happens in the drastic and the reflex is when you clasp your hand, your brain gets busy. It's busy doing other things like clasping your hand. And it forgets or lessens the stimulation to the Renshaw cells because it's busy doing other things. And since the Renshaw cells are not inhibiting as much, it's called disinhibition. So since the Renshaw cells are disinhibited, they don't attenuate the reflex as much, and the reflex response gets bigger. All right, so that's what we're looking at for that. So, kind of written out here, the drastic distracts the interneurons, and that's called cortical irradiation. That bigger reflex response has a scientific name. It's called cortical because it's cerebral cortex, right? We saw that increase in activity when we did the drastic. Irradiation just, basically means more activity, right? So more activity in the cerebral cortex, basically, is happening. When you do the drastic, your brain doesn't stimulate the Renshaw cells, which means we don't have those iPSPs to the motor neuron, which means more of the EPSPs get through, and that's why we see a bigger knee-jerk reflex, because we're stimulating more muscles from those excitatory stimuli that normally get canceled out by the Renshaw cell activity. But since Renshaw cells aren't as active, the reflex gets bigger. All right. So it's called disinhibition, and it's a nice physiological response that we see that we normally don't see other places. So it's a nice place to talk about it. Now, the plantar reflex is a little different. The plantar reflex goes through many synapses, and it's actually the longest reflex in the human body. It goes from the bottom of your foot literally to the top of your head and back down to the bottom of your foot. So it's a polysynaptic reflex. It's got many synapses. It's sometimes called a cortical reflex because it goes all the way up to the brain. Sometimes it's called a complicated reflex as opposed to a simple reflex that we saw with the monosynaptic and the knee-jerk reflex. So it's much more complicated. And what happens is basically you, by running down I guess it's up, probably a better way to say it, up the bottom of the foot, you set up the stimulation of the sensory neurons, and those go up the leg to the spinal cord, and then up the spinal cord all the way to the cerebral cortex, the brain, right? And then come back down through the cerebellum, down the spinal cord, out to the foot, and then give the response. So what we're looking for is a normal response. So we're looking to not see the Babinski sign. The Babinski sign is when the toes flare out or fan out. You see Babinski sign in infants because their nervous system's still developing. They haven't made all the connections yet. So that's normal. And you see it in adults where there's some damage along that pathway. But it could be anywhere along that pathway. So it could be a bad back, it could be a bad neck, it could be a tumor in the brain, it could be nerve damage in the foot, it could be lots of different things. It just says somewhere along that pathway there's some damage. And so they used to use the Babinski sign to assess spinal cord injuries. This is kind of sad, I guess. Maybe not so sad as a physiologist. One of my earliest childhood memories is watching a football game. And one of the football players ran into the other one and had a neck injury. And back then they showed everything, and they're a little more discreet now. And I remember them literally showing them taking the guy's shoes and socks off on the field, literally, before they did anything else, and then playing with the bottom of his foot, as far as I could tell. Now I know they were doing the plantar reflex to see if he had a Babinski sign. And the idea would be if he had a broken neck, right, then he would show a Babinski sign. Well, also, the problem is if he had not a broken neck, but an injured neck where he had some swelling, then he'd also show a Babinski sign. And so that practice has gone away from doing the Babinski sign, because even if he doesn't or she doesn't have a Babinski sign, then you're going to treat them like they broke their neck anyway, because they could have injury, and by moving their head wrong, you could make it much more severe. So you treat everybody like they have a broken neck, so it doesn't matter what you look at. And sometimes it's just the swelling, literally, that leads to a Babinski sign where the person still is normal. So oftentimes when you see people with spinal cord injuries, they don't know right away how much damage there was or how much function loss there was, if any, until they actually have a chance for the swelling to go down and do some other things to see if the person has movement and feeling, things like that. So that's kind of what we're looking at with the plantar reflex. So if you look at the spinal cord injuries, right, how would these different reflexes would be influenced at the spinal cord? So for the knee jerk and the plantar, right, what would happen at lumbar versus what would happen with the cervical? So if you injured your back below L4, the lumbar area, then the knee jerk would look normal. right? So that's below the level of the spinal cord where the knee jerk is mediated, so it would look normal. The plantar reflex, if it was below L4, you'd see a Babinski sign, and that's because the sensory neurons from the foot go in below L4. Remember, so L1 to L5. So they go in below L4, and we would see an abnormal reflex. We'd see a Babinski sign. The cervical one is the one that sort of messes students up. If we had a cervical injury in the neck, right? So if the neck was injured, the knee jerk would be enhanced. Why would that be? Well, normally during normal physiological happening, the brain is stimulating through the cervical area of the spinal cord, right, all the way down to the Renshaw cells, is stimulating the Renshaw cells. And the Renshaw cells, in turn, are opening chloride channels in the cell bodies of the motor neurons and inhibiting them. So if someone breaks their neck, the brain can't signal the Renshaw cells anymore, so they lose their inhibition. That's called disinhibition again. And so now more of the signals get through, and we see a larger reflex response. In addition, we'd see a Babinski sign, right, if you did the plantar reflex, because again, that reflex has to go all the way up to the brain and back. So we have to go through the cervical spine twice, once going up and once going down. And that's going to give us an abnormal reflex as well. So if you understand that, you sort of understand, I think, most of it in terms of those things. We will talk about one more lab, but it won't be in this lab quiz, so we'll worry about it later. So that's the reflexes for lab 1718. And again, the EEG, you don't have to know much, but the reflexes, some good, interesting physiology, and again, will be applicable for some of you that go into clinical medicine and nursing especially.